Wow. Thank you very much. It's so wonderful to see so many people here. When I was first asked to give this public lecture, it was easy for me initially to say yes, and then I had these pangs of anxiety. The first pang of anxiety was, would anybody come? And of course I thought, well, I could count on my mother, but then I realized she lived too far away and she probably couldn't make it. So then, of course, you think of your family. Well, my daughter, who's on the Hutch email list, because she likes to follow the science at the Hutch, sort of said, I just got to hear some Hutch hotshot speak. And I thought, no, okay, that's me. And then shortly after, I got this email. So I guess she's not coming either. So I'm very grateful to all of you. But I think what's really wonderful is to actually have the opportunity to communicate to the public some of what goes on at the center. I think sometimes we come into work early in the morning, we go home late at night, and we sometimes don't reach out, I think, to the people in the community. And as Rhoda said, our research enterprise here is largely funded by your tax dollars through the NIH. And so I think it's important for us to communicate to you what we actually do. So I didn't get to skip the homework part. So I'll actually, rather than send it home with you, I'm going to ask you if anybody, and I know there's somebody in here that knows this person, if anybody knows who this is. Yes? The Bubble Boy. This is the Bubble Boy. This is, his name was David Vetter, and he was what got me interested in medicine and in immunology. I was a high school student when I first read about the Bubble Boy. Some of you will know about him, the story. This was a boy that was born with a severe deficiency of his immune system, called severe combined immunodeficiency. Essentially, he had no immune system. And what his doctors realized at the time, because the family had had a previous child that had this disease, was they needed to protect him from the environment. I mean, with this disease, you essentially have no immunity to anything. Now, at the time when I read about this, I was a high school student. I lived in the wilds in northern Canada. I was outdoors all the time. And what struck me, first of all, seeing this young man in this bubble, where he essentially had no contact with the outside world, how challenging and how difficult that might be. And of course, there's been much written about the psychology of putting this child, and he spent many, many years in this isolated environment. But then what I realized is how important the immune system is and how little we actually understood about what it does for us. And that was what really motivated me. I will add that this severe combined immunodeficiency was later cured by bone marrow transplantation, largely developed here at the Hutchinson Center, providing you had a compatible donor, and now can be cured by gene therapy, where you actually take out the stem cells from the bone marrow, from the individual. You correct the gene by introducing the normal gene, and you can then transfer those cells back and essentially restore immune function. So that's the progress that's been made just since 1971 when David was born and put into this environment. But as I say, that's what really stimulated my interest in the immune system. So now we have to get into some of the science, because in order for me to go through this talk, you are going to have to understand just some of the basics of the immune system. So the immune system is really designed to do something very simple, and that is to distinguish self from non-self. And it does that through a complex network of cells and tissues. The immune system can be broadly classified into what's called an innate immune system, which are cells like macrophages and neutrophils. Any of you who've had chemotherapy will know about, they're looking for your polycount. When do the neutrophils come back? Those are cells that are immediate responders to an infection. And they're there for this rapid response. But then there is what we call adaptive immunity, which is a slower response, but has this wonderful property called immunologic memory. And these are the B and T cells that are in our blood. Now, it is those cells that David was deficient in, the adaptive immunity. So he was really unable to respond to viruses or fungi or bacteria that all of us sort of don't even think about. And it's these cells that I'm actually going to talk a lot about today, these T cells. So if we just think that what the immune system is trying to do is distinguish something, our own cells in our body, from something that's potentially dangerous to our body, that's a good concept to start with. Now, how do these T cells develop? Well, as I mentioned already, they do develop from the bone marrow. There's cells in the bone marrow that go to an organ in the chest called the thymus. And in the thymus, T cells now undergo a process of education where they learn to distinguish self. So if there's any T cells that have a receptor that would be reactive with the self tissues, those T cells are automatically deleted and they die. And only T cells are selected that have receptors that are tolerant to self. And now these T cells patrol the body and essentially look for non-self. They look for viruses that could infect cells or any pathogen that could get into the body. Now, just in case, the immune system is so amazing, just in case some T cell gets through that might be a little bit reactive with self and cause an autoimmune disease, we have these other cells called regulatory T cells that sort of keep the brake on these. And these turn out to be important because cancer cells actually tend to develop these regulatory cells in their environment. They sort of cloak themselves in these cells that might dampen a T cell response. Now, what the T cell sees is it sees fragments of proteins inside of cells that are displayed on the cell surface. So it's sort of like an early warning system. It's sort of like the alarm system in your house, right? If somebody starts to come in, breaks in, you're going to set off an alarm. Well, if a virus gets into a cell, it makes proteins that aren't usually made in that cell, non-self. And those can be displayed on the cell surface, and the T cell with the right receptor will see them and get activated to eliminate those cells. And so structurally, this is what it looks like. You have this receptor. Each T cell has a different receptor. You have these MHC molecules that display these small fragments on the surface of the cell. So a really highly orchestrated and evolved system. So I'm just going to go through some simple attributes of a T cell response to a virus because these ultimately become very important when we think about cancer. So the first thing is, if you think about it, a T cell might recognize now a cell infected with a virus. What does that T cell do? Well, it gets activated and it undergoes massive proliferation. So it then can kill all the virally infected cells. So as the virus is spreading, the T cell response is getting bigger and bigger and bigger and catching up and killing off these virally infected cells. And that's when you get a fever and you feel achy because these T cells are activated and they're fighting the infection. That's why your lymph nodes swell up when you get a cold or a flu. Now, once these T cells have eliminated the virus, then you end up with this wonderful thing called memory. Many of these T cells die, but you're left with some that are memory cells, and they last for the rest of your life. So how many in the room had a smallpox vaccine? Some of you guys are as old as I am. Yeah. So do you know that if I took a blood sample, I could still find smallpox-specific T cells in your blood now, and you will have those for the rest of your life. These memory cells have this incredible capacity for self-renewal and longevity. And that also becomes very important because if you think about it, wouldn't this be a wonderful system to treat cancer with? If we could target tumor cells, we could not only eliminate the cancer, but we could also have potentially immunologic memory against that tumor. So, can the immune system recognize cancer? Well, I have always found this quote to be just so amazing to me. This is Paul Ehrlich in 1909, who said, "I am convinced that during development and growth, malignant cells arise extremely frequently. But in the majority of people, they remain latent due to the protective action of the host." Now, what he was actually espousing here was a concept we call immune surveillance. And that is that we all are developing cancers, that some of our cells are making mistakes and becoming malignant, but that our immune system is actually recognizing those cells. Now, this was actually quite heretical at the time, but he actually went on and said one more thing. He said, "I'm also convinced that this natural immunity is not due to the presence of antimicrobial bodies, but is determined purely by cellular factors. These may be weakened in old age where cancer is more prevalent." Now, how prescient is that? I mean, isn't that amazing? In 1909, remember, he said this before T and B cells were even discovered. So he really, and I think this was one of the great things about scientists in those days, is that they didn't have the modern day tools that we have, but boy, could they ever think. And so that was really, I always found this to be incredible. But of course, there was no evidence for any of this. And so this essentially was a concept, this immune surveillance. It became actually, most scientists were very, very skeptical that this actually was true at all. But there is some evidence of that, and it actually, again, came from the Hutchinson Center. And that's called the graft versus leukemia effect of an allogeneic bone marrow transplant. Now, one of the things that the Hutchinson Center is famous for is, Dr. Thomas. I was very fortunate to come here when Dr. Thomas was active and when the program was really in its major growth phase, and to work with him, it was just an amazing experience. But what happened there is we were able to cure some types of leukemia by giving the patient high doses of chemotherapy and radiation, and then giving them bone marrow cells from a matched brother or sister. And we thought that it was the high doses of chemotherapy and radiation that were actually doing the job. But Paul Weiden started to analyze the data from the patients, and what he found was something very interesting, and that is if you did a transplant from a completely identical twin, so a syngeneic transplant. So this is two identical individuals, so essentially self, right? They're both self. The relapse rate of the leukemia was very high. But if you actually did the transplant from a brother or sister that wasn't completely genetically matched, they were HLA matched but not completely genetically matched, so there was some non-self there, now the relapse rates were much lower. And if you got this condition called graft versus host disease, which indicated a reaction of the donor's immune system against the patient's cells, the relapse rates were the lowest of all. So again, this was very, self, non-self. It really told us that the immune system could actually cure a cancer, and even cure an advanced cancer, because many of the leukemias in these days were very advanced. But still, the scientific community was skeptical that beyond this sort of unique situation of an allogeneic stem cell transplant, that the immune system would play any role at all in preventing progression of most cancers. And so most of us who, at the time, I got very interested, in fact, spent about a decade really trying to study this graft versus leukemia effect and really understand it, what is actually was being recognized by the T cells. Most of people in the scientific community really didn't think this was particularly worthwhile. They thought that because there was a revolution in molecular biology and we were now starting to map the mutations that cause cancer, that actually this was going to be the solution. That what cancer was caused by were mutation in genes that drove the proliferation, drove the growth of these cells abnormally, and abnormally maintained their survival. And that was what the problem was. And in fact, in lung cancer, we know there are mutations in KRAS. There are mutations in the epidermal growth factor receptor. So these are activating mutations that result in the cell undergoing proliferation and maintaining its survival. In melanoma, we learned there's mutations in a protein called BRAF that again activates a pathway that keeps the cells alive and gets them to proliferate. And in chronic myeloid leukemia, we understood there was a chromosome translocation that activated another kinase that made the cells grow and divide. And so everybody thought that all we had to do was essentially develop drugs that would target these mutations and we would be able to cure these cancers. And in some respects, that's been successful. In chronic myeloid leukemia, drugs that target the BCR-ABL kinase are incredibly successful in suppressing the growth of the leukemia cells, but they don't cure the disease. And in melanoma, the BRAF inhibitors work for about 6 or 7 months, and then the tumor comes back with a vengeance. So the cancer cells are finding ways around these things. So the fact that they've been driven to growth, they always find their way around them. But from the standpoint of an immunologist, from a T cell's perspective, the very fact that there are mutations, in fact, missense mutations that result in different amino acids, so different peptides, that means from the T cell's perspective, cancer could be non-self. And so for immunologists, this gave us the encouragement to actually pursue this even greater. Now, as we think about cancer, cancers are cells that are growing uncontrollably. And when cells grow, they make mistakes in their replication. And as the DNA replicates, they make mistakes. There is always some infidelity in the replication machinery. There are also many cancers result from mutations, from exposures to carcinogens, things like cigarette smoking and lung cancer, things like ultraviolet light and sun exposure and melanoma. So there are going to be lots of mutations in cancer cells, even those that aren't necessarily driving the cancer cell to grow. Now this is a paper that came out in Nature, and it's just dots, right? But all you have to think about is over here, this is the mutation burden per megabase of DNA. So there's about 3,000 megabases of DNA in a cell. And if you just look at the mutational burden, you can see that some cancers, like melanoma and lung cancer, where there is mutagen exposure, so carcinogens that are going to cause lots of mutations, they have lots of mutations. These are just individual cases and the number of mutations per megabase of DNA. So in some cases here, you have really hundreds, thousands of mutations in that cell. And in other cancers, you have few. Now, what's really interesting, again, from the immunologist perspective, is that one would then predict that these are the cancers that are going to be most susceptible to immune recognition. And as I'll show you in a few minutes, this actually turns out to be true. So cancers that were notoriously difficult to treat with chemotherapy or radiation therapy, because they're so mutated, and because they have all these resistance pathways to these conventional therapies, cancers like melanoma and lung cancer and now bladder cancer and head and neck cancer, these cancers are now turning out to be responsive to immunotherapies. And I'm going to come back later on to show you how we can genetically engineer cells to target some of these cancers down here where they don't have a lot of mutations. So again, from the standpoint of the immune system, the immune response to non-self would be predicted to be stronger out here than it would be down here. So is there any evidence, though, that the immune system even recognizes cancer? Well, there is. And again, this comes from sort of really good, what I call really good observational studies in human cancers. So if you actually look at colon cancer, and you look at the tumors, and look at just, are there T cells infiltrating the tumor? And you classify them into those tumors that have a high T cell infiltrate, either at the center of the tumor, at the invasive margin, or in both regions, and those that have low T cell infiltrates, and look at the survival. What do you see? The survival is much better if you've got a lot of T cells infiltrating your tumor. Again, suggesting that the immune system is actually doing something locally to control the growth of these tumors. But why doesn't it get rid of it? Right? If it's there, if it's seeing the tumor, and the cells are there and they're delaying its growth, why can't they eliminate it? Well, so I think I've hoped to convince you that the immune system is not an innocent bystander in cancer. The question then becomes, well, what is it doing, and why is it not able to eliminate it? Well, some really wonderful work was done by Rob Schreiber at Washington University in St. Louis, where he actually induced malignancies in mice with a carcinogen, with methylcalanthine, and it caused a type of cancer called sarcoma. And these were heavily mutated. And what he did was he really used this as a model to study recognition of the immune system. And he came up with essentially something that was quite interesting, and that is that early on, when you take normal cells and expose them to this carcinogen and they start to mutate, you actually get transformation, malignant cells arise, but those malignant cells actually do have tumor antigens. And in fact, many of them are eliminated. You'll get an immune infiltrate, and some of those will be completely eliminate the tumor, and you'll end up with essentially back to normal cells. All the cancer cells have been eliminated by the immune system. But more often, in fact, you'll end up with this equilibrium where there's some cancer cells still able to survive. The T cells are in there, but they're not really able to completely eliminate the tumor. There's a variety of reasons for that. Sometimes it's simply that the antigen that is on some of the cells, those cells get eliminated, but there are other cells that don't have an antigen. That's sort of what we call antigen loss or escape. But more often, it's actually a complex environment where the escape, where the tumor actually grows, but there's T cells that actually express molecules that normally regulate our immune response. It's when we get a viral infection and our T cells are massively proliferating, they can't do it uncontrollably. So they normally express molecules, things that we call them CTLA-4 or PD-L1. These are just names of molecules. They come up on the T cell that regulate their proliferation and growth. And it turns out what happens in the tumor microenvironment is all the T cells express these molecules, and they essentially get what we call exhausted or stunned, and they're not able to now proliferate and function to eliminate the tumor. So if we just go to that escape phase and think about this then a little bit, if these molecules like CTLA-4 and PD-1 are involved in this, what if we, and we know that they inhibit the T cell from proliferating, what if we block, I think we missed the slide there. What if we block them? And so what's happened is companies have developed antibodies that block CTLA-4 or block this PD-1, PD-L1 interaction, and that turns inhibited anti-tumor immunity, as I'll show you, into effective anti-tumor immunity. So the first landmark, really, was this study that came out in the New England Journal using a drug an antibody to CTLA-4. And this was done in metastatic melanoma. Now, you have to understand in metastatic melanoma, the survival rates are very poor. This is the survival of patients that actually just got a vaccine at the bottom here. And you can see that that with these patients from the time they enrolled in the study, the majority of these patients had died within 24 months. Now in this study, because they felt it was the first time they were testing the ipilimumab, they would actually give it with a vaccine, because the idea would be it may make the vaccine a lot better, because it would take the brakes off of T cells that were responding to the vaccine. And that really didn't happen. They did do better, but they did just as well if they got ipilimumab alone. So just blocking this checkpoint now was able to cure about 20% of patients. Now there was some side effects with this, because of course if you block this checkpoint, you might unleash some T cells that can be autoreactive, that could recognize some of your own cells. And so some of these patients got autoimmunity. They would get skin rashes, they would get a colitis. This was all readily treatable, but we had to learn how to treat it. But it was really, I think, the first evidence that said you could actually, with a single molecule that just interfered with one of these checkpoints, you could actually eradicate a cancer that was otherwise incurable. Now the same thing was subsequently shown with PD-1 blockade. This is in melanoma now with PD-1. This is just looking at individual patients at their tumors. You can see once they go on the treatment, instead of the tumor growing, the majority of patients had tumor regression. You can see very large tumors here regressing. Sometimes this takes a while because the T cells take their time in killing off the tumor. But the response rate in this trial with PD-1 or PD-L1 antibodies was about 40%. And again, in lung cancer, another highly mutated tumor, we see that we have wonderful responses where we see shrinkage of the tumor. Here you can see metastases. Here 15 months later, they're gone, metastases in the liver. So this told us again that these two checkpoints, really just targeting those individual molecules, could unleash an immune response in the body. It was already there. It just needed to be let go and eliminate tumors. But of course, not everybody was responding. So what about if you combine the two of them? So this is the next big breakthrough. This is actually combining now anti-CTLA-4 and PD-1 in here. And you can see now almost all of the patients respond. And they get deep responses. So you can see many of these patients have 100% reduction, what we call a waterfall plot. So this is just the change in size of the lesions. You can see many of these patients have 100% reduction in their tumors. And these remissions are quite durable. They're lasting long periods of time. Many of these patients appear to be cured, just by letting the immune system that's in the body do its work. So what have we learned from these checkpoint blockers? Well, we've learned that this takes the brake off. It allows the endogenous immunity that's there against these tumor cells to recognize them and eliminate them. It tells us, I think, that cancers can be eliminated by immunotherapies, and that this is actually a very powerful therapy. It doesn't require chemotherapy. It doesn't require radiation. It's just letting cells that are in the body do the job. Now, it doesn't work in everybody, and not all tumor types respond to this. And so there are many, many questions that are really the subject for ongoing research now to really improve this. There are other checkpoints that I'm not going to talk about that are being actively pursued. These agents are now really in the realm of big pharmaceutical companies, and they're developing them quickly and rapidly. These are some of the questions that are being pursued, and they're doing it in conjunction with scientists at places like the Hutchinson Center, where we actually have the technologies to really delve into this deeply and try and understand how we can improve this. Now, what about patients, though, that don't respond, or patients at the other end of the spectrum that don't have a lot of mutations? And so one of the areas that we've worked on for many years at the Hutchinson Center, and this is work that really started with Phil Greenberg and I, shortly after I came here, when we started to try and reconstitute immunity after transplant to a virus by taking T cells from the donor that were specific for that virus, growing them up in the laboratory and transferring them. And so the question that we've really evolved to, that worked, by the way, but of course treating viruses, usually you can eventually develop drugs that work for those viruses. But the question that we really wanted to get at was, could we actually isolate or potentially even genetically engineer T cells? So make an immune response outside of the patient's body from their own T cells that we took from them to recognize and kill their tumor cells. So I want to talk a little bit about the first iteration of this, and we now have the only program, I think, on the West Coast that's doing this. It's called Tumor Infiltrating Lymphocyte Therapy, or TIL therapy. And this is work that's being led by Sylvia Lee in our program at the Hutchinson Center. This was originally developed at the surgical branch at the National Cancer Institute by Steve Rosenberg. Steve was really a pioneer in cell therapy, and I think had the same kind of determination for TIL therapy that I think Dr. Thomas had for transplant. But the idea here is that if you take a tumor fragment, and this was initially done largely in melanoma, although now we have interest in doing this in all of those cancers that have a high mutation burden. But if you take fragments of the tumor and actually plate them out in the laboratory and culture them in interleukin-2, just a growth factor for T cells, and then you screen individual wells, you can find some wells that will actually recognize the tumor. And if you expand just those cells, and you can expand these cells, T cells are wonderful. They proliferate like crazy, so you can grow billions of these cells. If you expand them to billions, then you can give them back to the patient. And this is called adoptive T cell therapy with tumor-infiltrating lymphocytes. This is some of the data from Dr. Rosenberg's work early on. And as you can see, these are patients now that have metastatic disease. This is a lung nodule, metastatic melanoma in the lung. This is a huge mass in the abdomen. 5 years later, this patient has no tumor at all. This is another patient. You can see all of these holes in the liver. These are all metastases, melanoma metastases in the liver. And you can see 6 years later, this patient has no tumor left. So the response rates with TIL therapy, when you can grow TIL that react with the tumor, are about 50 to 70%. And about 20% of these patients have durable, complete remissions. And many of these patients are now more than 8 years after their therapy. So in the cancer field, if you're out more than 5 years, you sort of cautiously say you're cured. So this is a very important proof of principle, but this is a really cumbersome therapy. It's really hard to do. I think it's important as a scientific tool because we can start to understand what are the antigens, what are the peptides that are actually being recognized by these T cells that are being displayed on the cancer cells. Are there common antigens in different patients' tumors that we could target? And so we're using this really both as an option for patients, but also as a tool to discover and improve the therapy so we can ultimately make it better. Now, I just want to point out that this TIL therapy works even in patients that have failed ipilimumab, the anti-CTLA-4 antibody that I talked about. So it's not that the checkpoint blocker will rescue everybody that has an immune response. That's not true. Some of those patients will still progress, and you can generate TIL from them, and you can still cure a large fraction of those patients. Now, the other area, though, and the area that we've really focused on, is this idea of being able to genetically engineer T cells. And this now will go across many, many different tumor types. And the idea here is, well, if it's the T cell receptor that recognizes the antigen, we could take any T cell from the patient. And if we had a library of receptors that recognize cancer antigens, and this library of receptors are going to be encoded in what we call gene transfer vectors. We could essentially then take this T cell, expose it to a virus that would deliver this DNA into the cell, and express one of these receptors, whatever the tumor type was in the patient, to target the cancer cell. And so you could actually now essentially make these cells very, very rapidly, not have to wait weeks to grow them out of the tumor infiltrate, but you could actually just take a blood sample from the patient, engineer the cells, and essentially propagate them up. And Phil Greenberg's lab has been working on this, and they have clinical trials already in acute myeloid leukemia targeting a cancer antigen that's now being extended to lung cancer, and the lung cancer trial, I think, will open in the next few months. So this is something that is not just science fiction. This is real world, and this is something that we will be able to offer our patients very shortly. Now, will it work? The answer is it will probably have some activity, and we're going to have to improve upon it. And that's where the research comes in. It's understanding what are the right cells to put this in. And so should we put it into memory cells that are already programmed for longevity, or should we put it into other types of cells? So my own lab has been actually focused on synthetic biology. So the idea being that, well, why can't we create receptors that would target molecules on cancer cells? And this opens up, I think, a completely new area of science. So now what you do is you take patient T cells and you design a receptor that has a component on it. Usually this is derived from a monoclonal antibody, so an antibody that binds to something on the tumor cell. But you encode that in a piece of DNA that would attach it to signaling molecules that would signal the T cell. Now this is actually something that, again, it sounds sort of like science fiction, but as I'll show you in a few minutes, it's something that's already in patients and having dramatic effects. So what do we do here? Again, we simply engineer the patient T cell to express this receptor, and it will now target the cancer cell. So how does this work? Well, first I'm going to show you how it works in a test tube. So these are T cells. You can see them here. I just should point out the T cells are these little guys here. They look like hand mirrors, little translucent things. And the tumor cells are all these big grungy tumor cells. This actually happens to be a brain tumor. And these T cells are engineered with one of these synthetic receptors that would target a molecule on the surface of this brain tumor. I'm going to show you what happens when we start this movie. And you just watch those T cells run around and come up against the tumor cell, and that tumor cell disappear. Okay? And pretty soon, the tumor's gone, and you have these clusters of T cells. That's how powerful they are. They simply eradicate the tumor that fast. And you can do this over and over again. Now, this is in a dish, of course. And that, you may say, well, that doesn't mean very much. And I'll show you how we move this from the dish to a mouse and eventually to a person. So the only thing the T cells say at the end of this is, what's the main course, right? So the first effort to use these synthetic receptors is to target a molecule called CD19. Now, CD19 happens to be a molecule that's on all B cells. Okay, so B cells are cells of the immune system. They're the ones that make the antibodies. So this isn't a molecule that's only on the tumor. It's actually on normal B cells as well. It comes on when the stem cell decides that it's going to become a B cell. But of course, because it's on in all B cells, it's also on in the leukemias and the lymphomas that come from B cells. So it's on acute lymphoblastic leukemia, Burkitt's lymphoma, mantle cell lymphoma, chronic lymphocytic leukemia, follicular lymphoma, diffuse large cell lymphoma. There's about 80,000 patients diagnosed with these diseases in the United States every year. So what has happened is that if you can engineer, using an antibody to CD19, you can now engineer a T cell to target these malignancies with T cells. And so these are experiments that Daniel Sommermeyer and Paula Kazasi in my lab have done. The first thing was to test this out in a mouse. Now what we did, interestingly enough, is we took a David Vetter mouse. So this is a mouse that actually has no immune system. It's what we call a Nodskid gamma chain mouse. It lacks all T cells and B cells. Now, why is that mouse any good? That mouse is good because it has no immune system, and so we can put human cancer cells into that mouse, and they will grow. We can put a human immune system into that mouse, and it will work. So what we do is we take that mouse and we engineer the lymphoma cells, in this case, with a gene from the firefly called luciferase. And the reason for that is that then we can actually, using light, we can image the growth of the cancer in the mouse. So we inoculate the lymphoma, and you can see the lymphoma starts to grow. And then we can treat those mice with CAR T cells that have one of these chimeric receptors that target the lymphoma, or with T cells that lack it. So if you give the mice controlled T cells, this is day 6 before the mice get there. You can see the tumor here, but it just keeps growing. And by 20 days, the mouse is full of tumor, full of luciferase cells. If we give them, though, CAR T cells from a certain type of memory cell, you can see it has an anti-tumor effect, but it doesn't cure the mouse. But if we use these, what we call central memory T cells, these T cells that are programmed for very long longevity in a person, they will actually work and eradicate all of the tumor in the mice, and you can cure these mice. So this really suggests that this is a very powerful therapy that we could potentially use for patients. So we've then taken this now from mouse to man. This is work that's being done by David Maloney, who's a lymphoma expert, and by Cameron Turtle, who's trained in my laboratory in T-cell biology and is also an oncologist that looks after these patients. And he's really coordinating all the immunological monitoring on the study. And we're treating now 2 types of cancer, acute lymphoblastic leukemia and non-Hodgkin's lymphoma. Now, the way we do this, it's not so complicated. We take out a blood sample from the patient, we do a genetic modification, we culture the cells. It takes us about 2 weeks to engineer and culture the cells to grow up enough to give the patient. Now, the reality is we can actually do this in about 5 days now. But in this trial, we're doing it in 2 weeks. And then those cells are reinfused into the patient. Now, of course, in these kinds of trials, you have to treat patients that have failed everything else in the beginning. So this is a patient who had lymphoma, and he had 3 different types of chemotherapy over several years. He'd had an autologous stem cell transplant after very, very high-dose chemotherapy, and his disease still recurred. And this is just a CT scan of his neck showing these very, very large lymph nodes. This is the dose of T cells that he got on the trial. Okay? Now, that is a pencil tip. You can see this is a very, very tiny number of T cells. And we think that if these T cells are the right T cells and they're programmed appropriately, that they will be able to, in fact, grow in the patient. And so we don't have to grow them for long periods of time in culture. But this was the dose that he got. It was a dose of about 20 million cells. This is what his scan looked like a month later. You can see dramatic regression of these massive tumors with that tiny dose of T cells. Now, why does it work that way? Well, it works that way because this is a living therapy. These T cells, as I showed you before, just like when they see a virus, a few become many, and they proliferate dramatically. And they'll proliferate until the tumor is gone. And so, because of that, we don't need to essentially grow billions of cells like we do with the TIL trial, because these cells can actually be engineered to proliferate in this way. This is another patient with a lymphoma. This is, again, a PET scan now. Tumor cells eat glucose, so we inject some radiolabeled glucose, and it goes right where the tumor is. And it lights up this big mass in the liver. This is also a patient who'd failed a transplant, failed many cycles of chemotherapy. 30 days after CAR T cells, after the first dose, he still has a tiny little tumor there. This patient is just about a month after his second CAR T cells now, and we're anxiously awaiting to see whether the second dose of cells will get rid of that last little bit of tumor. Now, what about acute leukemia? This is another PET scan of a patient who failed an allogeneic transplant, failed that graft versus leukemia effect. This is a PET scan. All of the bones in this patient light up. This is how much tumor the patient had. If you look in their bone marrow, all of the cells in the bone marrow, these little red dots, each one of those is a leukemia cell. 84% of the patient's cells in their bone marrow were leukemia blasts. So just an enormous tumor burden. The reason I circled this here is these are the kidneys. Now, normally when you give the PET scan, the F-18, the imaging glucose, it actually accumulates in the renal pelvis, this area here, because it'll go out in the urine. This isn't. It's actually in the kidney. The leukemia was infiltrating this patient's kidneys. That's how much tumor she had in her body at the time we treated her. And in fact, her kidney function was deteriorating very rapidly. We were actually worried we would not be able to treat her. She got, again, that little tiny dose of T cells. And this is a month later, actually 37 days later. You can see the bones now are normal. The kidneys, now the glucose is in the right place. It's being excreted in the urine. The kidneys are normal. And if you look in the bone marrow, there's none of these red dots. The bone marrow is completely normal. Every leukemia cell has been eradicated. Now, that is remarkable. And I think in my career in science and in medicine, I think these are amongst the most dramatic responses that I have ever seen with any therapy. Now, it's not perfect. When you eradicate a large tumor burden like that with those T cells are proliferating. They are going crazy in the patient. They are producing cytokines. And we see a syndrome, especially in the patients that have a large tumor burden, called the cytokine release syndrome. These patients get high fevers. They feel like they have a terrible flu. They can get, much like you when you get really sick with a flu, they're really in bed, and they can get very, very sick. We call this the cytokine storm, and some of our patients have taken to T-shirts that says, "I survived the storm." The patient actually came into clinic with that, which really pleased Dr. Maloney, I can tell you. But it is a serious problem, and we're learning how to manage it. And I think that part of it is, as we treat patients with lower tumor burdens, we won't see this severe effect. Right now we know that if we treat a patient that has a high tumor burden with a high dose of T cells, we're going to get a very severe cytokine storm. But if we treat a patient with a low tumor burden, we can give them a lot of T cells, and the leukemia will go into remission without many side effects at all. So part of it is going to be selecting patients and treating them at the right time. So this is just some of the data from Dr. Maloney and Dr. Turtle. In ALL, the complete remission rate. Now remember, these are patients that have failed everything. The complete remission rate is 83%. In lymphoma, the response rate, where we have both complete and partial remissions here, is about 60%. Very clearly, eradicating every last tumor cells with these big lymph node masses is more challenging. We have to continue to work on that, and we have a variety of strategies that we're taking forward. But the survivals are excellent. for a group of patients that really probably, to be honest with you, only had months to live. So based on this, the work that I've shown you on checkpoint blockers and on adoptive T-cell therapy, in Science magazine labeled Cancer Immunotherapy as the breakthrough of the year in 2013. And the little title in here is T-cells on the attack. And of course, for those of us who have been in love with T-cells for many years, that's very gratifying. But we have a long way to go. We have a long way to go. So the question is, with these chimeric antigen receptors, with these synthetic receptors, can we go beyond these leukemias and lymphomas? Can we target common solid tumors? And so we have a project in the lab that, and I see Ashwini's who works on this project, we call it the Limbersky Project. And the reason we call it the Limbersky Project is because Mark and Raylin Limbersky, about 5 years ago, I met with him actually just to talk about what was going on in my lab. And he said, what do you have that you would really want to do that, that you're not doing? I said, well, we have this protein called ROR1 that we're really interested in. It's on a certain type of leukemia. And I'm really interested in looking at whether we could target it with one of these chimeric receptors. And so they have supported us for the past 5 years with, I think they've supported us financially, they've supported us emotionally at times, because this has been a hard project, to actually get this to the point where we're ready to go into the clinic. Now, what were the challenges? The first challenge was you have to identify a target that's only on cancer cells and not on normal tissues, and ROR1 seems to fit that bill. So it's on many types of cancer, CLL, a type of leukemia, lung, breast, melanoma, many types of cancer it's expressed on. It has minimal expression on normal cells or no expression at all. There's a couple of normal cells, normal tissues that have low level of expression, but we don't think that's going to be a problem. We've designed and optimized receptors. This has been a process that's taken us years to do, but we've optimized receptors. We've done safety testing now in animals that have the same expression pattern for ROR1. And we think we're going to have to do some things to overcome the solid tumor microenvironment because there are many other cells there that suppress immunity, and we're working on models that will allow us to do that. But we already have data with the ROR1 CAR T cells to demonstrate that, for example, that they will eradicate a breast cancer. So now this is a luciferase-labeled breast cancer going into a non-skin mouse, a human breast cancer, and we're treating them now with the ROR1 CAR T cells. You can see with our very best CAR, these are the tumors in the control mice that progress. With a ROR1 CAR that's sort of suboptimal, you have an anti-tumor effect, but you don't cure the mice. But with this, what we call the R12 CAR, we cure almost all of the mice. So this is now going into a clinical protocol. The FDA IND application will go in early May, hopefully, and hopefully we'll be treating patients with ROR1 positive cancers by next fall. And I think for us, this is very exciting, and I think for the Limberskys, something that they can be very, very proud of to have helped us get this far. But how far can we go with this whole concept of synthetic biology and CAR design? So I want to talk a little bit about a project, and again, Ashwini works on this project, and I sort of call it making molecular Velcro to treat cancer. So if you think about it, what we're really doing with these synthetic receptors is we're taking a protein that is able to bind to the cancer cell. And so you might imagine that there's all sorts of things you could do. And the cancer cell surface now essentially becomes your playground, right? You can sort of say, we can start to look at things that would bind to the tumor that wouldn't bind to other things. And we've been collaborating with a colleague of mine at the University of Zurich in Switzerland who looks at proteins called designed, anchored, repeat proteins. And these proteins are really cool because they have this binding interface. They're normally made to interact with other proteins. And he's made libraries of these, very, very large libraries containing millions of molecules. You can actually screen them to find proteins that would bind to your target of interest. And so, for example, just to prove the principle, Ashwini in the lab started to try and see if she could design a DARPin CAR that would bind to the epidermal growth factor receptor. Remember I told you that that's upregulated in many cancers. And so this is where there's actually an antibody that's used in the clinic called cetuximab, and this is where it binds. And we've designed a DARPin that binds in a very similar region to cetuximab. It actually has an affinity that's probably a little bit better than the antibody. And if you actually make this DARPin into a CAR like this, you can actually now treat, and now here we have a breast cancer that expresses high levels of EGFR, and you can see that with the DARPin CAR, you can actually eradicate the breast cancer, whereas the control cells don't eradicate the breast cancer. So we think that with this molecular Velcro, we can start to go after all sorts of things on cancer cells that we couldn't approach before. And I think the future really is going to be this combination of the cell science, understanding the cells that we put into patients and understanding the targets that we go after. So I just talked about the tip of the iceberg in cancer immunotherapy. I talked about immune modulatory antibodies and adoptive cell transfer. All of these other modalities are being tested in various labs around the country. We are entering, I think, a new era where immunotherapy is going to become as established as surgery, radiation, and chemotherapy. And I'm hoping that it's going to be a lot safer for patients and a lot more effective. And we'd just like to mention that, of course, I don't do this work myself. I'm very lucky to have an amazingly talented group of people in my laboratory, as well as in the program. I could have put up, a portrait of our entire program. There's 50 people in the immunology program. And I'm very, very lucky to be at the best cancer center in the world. So with that, I'd like to thank you very much for your attention.